Hi everyone. Today we're gonna solve this paper by Mock Championship by Fastpapers, and this is GCE Biology paper 4, alternative to practical, and the total mark is 40. Glucose is a reducing sugar, and Benedict solution is used to detect reducing sugars. Describe the procedure you would follow to test a sample of fruit juice for the presence of reducing sugar using Benedict solution. Include an appropriate safety precaution in your answer. So there's 3 marks here, and you use drops of Benedict solution into the fruit juice and heat the mixture. So for mentioning heat, you get 1 mark, and use a temperature regulated water bath. There is another mark here to control the level of heat, and use eye protection while observing color change. So there can be a lot of vapor, and just to get an eye protection for this is necessary. Some students were provided with 1% glucose solution and distilled water. They diluted the glucose solution to obtain 5 solutions of varying concentration. Each of these solution was then tested using Benedict solution. The concentration of the prepared solution and the corresponding test results are shown in table 1.1. So we didn't have this value. This is a blank one, and we have this. The student also decided to test the distilled water with Benedict solution. Write the result of this test in table 1.1. So the original color is blue, and there is no glucose concentration, which is 0 here in distilled water. So this is gonna be blue. Okay, explain why students tested the distilled water. So obviously this is used as a control because you want to check the color change of Benedict solution that is affected by the presence of glucose. So in distilled water, since there is no glucose, that is used as a control. The students compared the color of their solutions after the same length of time. State 3 other variables that the students should have control to make their results comparable. So other than the length of time, what else should be the control? And it should be the volume of solutions used. So the volume of fruit juice should be consistent, and the volume of Benedict solution, or it also can be concentration. So you mention either volume or concentration to get the mark. And temperature, obviously, just so that there is valid comparison. The students were then given a glucose solution X with an unknown concentration. They repeated the Benedict test using the same method and observed a yellow-orange color. So in D1, using the results in table 1.1, state the likely percentage concentration of glucose in solution X. Explain how this conclusion was determined. So since we have yellow-orange in the question over there, it should be somewhere over here, right? So between yellow and orange. So this is 0.3, this is 0.4, and we can safely assume 0.35 or any number in between the two. Okay, so what is the explanation? It is a color between yellow and orange, where yellow indicates 0.3% and orange indicates 0.4. So we choose a mid value, which is 0.35. Then the student found it difficult to decide the exact color of solution X after testing it with Benedict solution. So suggest a reason for this difficulty and a method which could be used to improve their confidence in deciding the color. So they're having some issues in deciding the color, right? So a little bit emphasis on the question and what it's saying. So the exact color may have been differently concluded by different students. So the interpretation of the results can be subjective, or it's possible that the color was difficult to describe in general. So you can mention either of these points to get a mark here. And the improvement is depending on what you mentioned here. So for the subjectivity of it, you use one student to record all colors, so the judgment is same across all the observations. Or if the color is difficult to describe, you use a color chart. Okay, here describe how students could use a 1% glucose solution to produce 5 centimeter cube of 0.5% glucose solution. So you want to essentially make the concentration half. So what you do is you take half of this volume you want to get of 1% glucose solution, and then add another half of the same amount of water to make 5 centimeter cube of 0.5% glucose solution. So you're using 1% and you're diluting it with water to make the concentration half. After testing the glucose solution with Benedict solution, the students observed that a solid had formed at the bottom of the test tubes. They concluded that measuring the mass of this solid could provide another way to estimate the glucose concentration. So suggest how the solid could be separated from the solution and how its mass could be determined. So a very simple answer. You use a sort of filter to extract the residue, and then you measure the mass of the residue. Okay, so in question number 2, plants can be provided with fertilizer to help them grow. Some students were provided with germinated seeds of the same species in some shallow dishes and a 10% fertilizer solution. One dish is shown in figure 2.1. So this is a dish over here, and these are germinated seeds. And then you have to identify the independent variable, which is obviously the fertilizer concentration. And the dependent variable is the height or the mass. So either one, because that's the one you're measuring, because that's the outcome of the experiment. So identify one control variable in this investigation. So the slash indicates either point, so any one between the slashes gets you 1 mark. And for this one, it can be same light intensity, carbon dioxide levels, temperature or volume of water. So basically you mention this, or same either of these points. Okay, so this is the last one. Lemna is a small green plant that floats on the surface of water in ponds and lakes. It consists of leaves which float and a root that hangs down in the water. So figure 3.1 shows a single plant that has 4 leaves. D and E indicate the maximum length of 2 of the plant's leaves. So this is the distance that we have to measure here. And figure 3.1, draw a straight line and measure the length. So since it's in millimeters, you convert centimeter to millimeter, and then you do the magnification calculation, which is image divided by the actual length. So in here we have 12 times magnification indicating here, and the length we measured was 48, so we put 48 here, and then we divide 48 by 12, which is 4, and this is the actual length. The population of this plant grows by each plant dividing into 2 smaller plants. These smaller plants then grow new leaves and divide again. Some students decided to investigate the growth of Lemna plants. They placed 6 plants in a small beaker containing nutrients in distilled water, making up a nutrient solution, and they used a lamp to provide constant light. Okay, so suggest why the students added nutrients to the distilled water. So obviously nutrients allow a better growth, so you just simply state that it's necessary for growth or just to have an improved growth. Okay, the students decided to measure the growth by counting the total number of leaves in at the same time each day at the start of the investigation. There were 16 leaves in total of the plant, on the plants. Figure 3.2 shows the beaker seen from above on day 4. So what you need to do is you need to count the total number of leaves visible in figure 3.2. So you count each of these things. So the total, the whole of this is not one leaf. It's obviously you need to count each subsection, and the total is 42. And in here, draw a line graph of the data shown in table 3.1. Join the points with ruled straight lines. So this is not a line of best fit. You just rule a line from one point to the other, and it has to be straight. So in y-axis we have total number of leaves, and in x-axis we have time, which is measured in days. So it starts from 0 and then we go up to 2, and now you have to calculate the number of leaves would have been present on day 1. So you draw a line from 1 and you put it here, and this is clearly 18 because one box is total number of leaves on day 1, 18. Okay. So predict the shape of the graph after day 7 if investigation continues for another 6 days, and you explain your answer. So the prediction is the total number of leaves will increase at a slower rate, and I added this line just for explanation, but this is enough. So as the gradient is already decreasing from 6 days, so this is a steep increase, right? But from 6 to 7 the increase is not as steep as it was. So we can predict that the growth rate is gonna the rate of increase is gonna slow down. Okay, so the explanation is plants could run out of space, or they could run out of nutrients, and also the water surface could be filled up, so there wouldn't be any room for them to grow further. Okay, so these are the last 2 questions. Suggest one other method that the student could use to measure the growth of Lemna. So since you're counting the leaves, now you could use a different method, and you could measure the mass of the plants at the end, or you could calculate the surface area of leaves. So lastly, plan an investigation to determine the effect of different concentrations of a nutrient solution on the growth of Lemna. Use the same method of counting the number of leaves that the students used in their investigation for measuring growth. So for each line here, I mentioned a couple of points, and for 6 marks you need to mention any 6 here. So firstly, take a range of 5 different nutrient concentrations, which is the independent variable. So the mark scheme says that you need to have at least 3. So just to be safe, I wrote 5. And then use the same volume of nutrient solution for each of the 5 concentration. So even if the concentrations are differing, like in 5 different levels, you need to use the same volume of the nutrient solution. And then ensure that there are the same number of plants or leaves at the start of the investigation. So just for consistency and so that the measurement is accurate. Keep the temperature and light intensity constant for all concentrations. So for these 2 control variables, you can just mention 1 to get 1 mark for mentioning control. So and then count and record the number of leaves at regular interval. So and give an example such as at the same time every day. Repeat the procedure at least 2 times for each of the concentrations and calculate means. So the average value will obviously give you a better explanation of the trend of the increase. And then compare growth or number of leaves at different nutrient concentration over time to find the rate of growth. Sorry, we did a line graph, my bad. You can do a bar graph for the rate of growth for different concentrations. So you could mention the concentration is at any specific level, let's say 0.1 for example, and you have 5 other concentration, and you could put a bar graph, but you could also use a line graph. Whichever feels suitable, you will get a mark anyway with anything you mention, but the graph is the marking point here. So the bar graph is just an example. Line graph is also acceptable. So anything is okay here. So this is all for this paper. Good luck for your exam.